close your eyes for a moment and imagine standing on a beach. Not a beach you have ever seen. Not a beach from any photograph, any memory, any dream you have carried with you. The sand beneath your feet is red, brown, and iron-rich, stained by chemistry rather than by life. The air you would breathe, if you could breathe it at all, holds barely a trace of the oxygen your lungs require. The for you is vast, immense. Before you is vast, immense, immense, perhaps more extensive than any sea that exists today. The surface catches the light of a sun that appears much as it does now. And yet the day is shorter than you have ever known. The earth turns faster in this era. And the moon. The moon is closer here. Measurably, visibly, perhaps disturbingly closer. It has not yet drifted to its current distance. It pulls the tides with greater force. Those tides surge and retreat in patterns we would not recognize. This is the Proterozoic, specifically, the Middle Proterozoic. A span of time so vast it dwarfs all of human history the way the ocean dwarfs a single raindrop. We are standing, in our imagination, approximately 1 billion years in the past. What you see instead, in the shallows, where light penetrates the water, are structures, low, rounded, layered. They look almost geological at first glance. But they are not geological, they are biological. Communities of microorganisms building themselves slowly upward through the patient accumulation of thin mineral layers. These are stromatolites. And in this moment, 1 billion years ago, they are among the most important organisms on Earth. They have been building for billions of years before this point. They will continue building for hundreds of millions of years afterward. If you turn from the sea, you would see the land. And the land is what, perhaps more than anything else, signals how completely alien this world is. There are no trees, no grass, no lichen, no moss, no soil in any sense we would recognize. The rivers that cut through this landscape carry sediment into the sea, just as rivers do today. But the riverbeds are bare, the banks are bare, the plains beyond are bare. The colors are ochres and reds and grays. The colors of iron-bearing rock, of quartz, of minerals shaped only by physics and chemistry with no life to soften them. It is beautiful in its way. A terrible ancient beauty that belongs to no human category of landscape. This is where we begin, 1 billion years in the past, on the shore of a vanished world. Geologists call this interval the boring billion. Do not be misled by the name. It was coined with a certain dry scientific humor, an acknowledgement that from the perspective of geological drama, the interval appears curiously stable. But within that stability, the foundations of everything we are were quietly being laid. The banded iron formations. Those ancient striped rocks of red and dark gray that miners today extract for steel, were deposited when dissolved iron in ancient seawater reacted with oxygen produced by photosynthesis. Life made those rocks. In layers, across hundreds of millions of years, life wrote its chemistry into the stone. The oxygen that fills your lungs right now, every molecule of it, was produced by life, not by geological processes, not by chemistry without biology, but by living organisms, by photosynthesis, by the patient work of organisms like those cyanobacteria, repeated across billions of years. We are not observers of deep time. We are participants in it. The mitochondria in your cells right now are the descendants of those ancient bacterial symbionts. The carbon in your body has been living for billions of years. You are the heir to 1 billion years of patient work. Close your eyes for a moment and imagine standing on a beach, not a beach you have ever seen, not a beach from any photograph, any memory, any dream you have carried with you. This shore belongs to a world so old, so fundamentally different from our own, that every category you use to understand the earth must be set aside before you can begin to perceive it. The sand beneath your feet is red-brown and iron-rich, stained by chemistry rather than by life. The air you would breathe, if you could breathe it at all, holds barely a trace of the oxygen your lungs require. There is carbon dioxide here, methane in thin wisps, nitrogen in abundance, and a faint sulfurous thread carried off the sea. You would lose consciousness within minutes. And yet, by the standards of this world, the atmosphere is doing exactly what it should. The ocean before you is vast, immense, perhaps more extensive than any sea that exists today, because the land is gathered differently here, bunched together in ways that leave enormous open stretches of water unbroken by any coast. The surface of that ocean catches the light of a sun that appears much as it does now, and yet the day is shorter than you have ever known. The Earth turns faster in this era. A full rotation takes perhaps 18 or 20 hours. The sun does not so much cross the sky as rush across it, and the moon is closer here, measurably, visibly, perhaps disturbingly closer. It has not yet drifted to its current distance. That drift is slow, a few centimeters per year across geological time, but 1 billion years of slow accumulation places it noticeably nearer to the horizon, slightly larger in the sky, pulling the tides with greater force. Those tides surge and retreat in patterns we would not recognize. They are not the gentle twice-daily rhythms of a modern shore. They are stronger, wider, more restless. This is the Proterozoic. Specifically, this is the Middle Proterozoics, a span of time so vast that it dwarfs all of human history the way the ocean dwarfs a single raindrop. We are standing, in our imagination, approximately 1 billion years in the past. Some geologists call this interval the Mesoproterozoic, others more poetically, and not without some scientific justification, call it something else entirely. We will come to that name in a moment. What matters first is this. You are looking at a planet that has already been alive in some sense for more than 3 billion years. Life began here long before this moment. Microbial life, ancient, tenacious, slow, has been threading its chemistry through the oceans for longer than the mind can comfortably hold. And yet, standing on this shore, you would see no fish in the water, no birds in the sky, no insects, no plants, no animals of any kind. Not on the land, not in the shallow bays, not in the tide pools. The tide pools themselves exist. The rocks are real, the water is real, the chemical processes are real and ancient and complex. But the visible, moving, recognizable life that we associate with Earth, the life that has fins and eyes and roots and leaves, that life has not yet arrived. What you see instead is this. In the shallows, where light penetrates the water and the seabed is shallow enough to catch the sun, there are structures, low, rounded, layered. They look almost geological at first glance, formations of sediment perhaps, or something deposited by a receding tide, but they are not geological. They are biological. They are communities of microorganisms, primarily cyanobacteria, building themselves slowly upward through the patient accumulation of thin mineral layers. Each layer is a season or a cycle or some rhythm of growth we can barely reconstruct. Each structure is ancient beyond all telling, and yet it is alive. These are stromatolites. And in this moment, and 1 billion years ago, they are among the most important organisms on Earth. They have been building for billions of years before this point. They will continue building for hundreds of millions of years afterward. Some of their descendants still exist today in sheltered hypersaline lagoons in Australia and the Bahamas, growing with geological patience in waters too salty for most predators to enter. When you look at those living stromatolites today, you are looking at something that connects directly to this shore, this moment, this vanished world. But here in the Middle Proterozoic, they do not share the sea with algae mats or jellyfish or anything complex enough to eat them. They are nearly alone in the shallow waters, doing what they have always done, capturing sunlight, fixing carbon, releasing oxygen in incremental quantities that will eventually, very eventually, change the atmosphere of the entire planet. We can only imagine what this shore smelled like, mineral and salt and something faintly sulfurous, perhaps. We can only imagine the quality of its silence. No wind through leaves, no calls of birds, no surf wash carrying the distant sound of life. just the water and the rock and the slow chemistry of a world still becoming. Somewhere behind you, if you turned from the sea, you would see the land. And the land is what, perhaps more than anything else, signals how completely alien this world is. There are no trees, no grass, no lichen clinging to the rock faces, no moss softening the edges of boulders, no soil, at least not in any sense we would recognize. Soil, as we know it, is made by life. It is a community of organisms, a matrix of decaying organic matter and minerals and fungal threads and bacterial colonies. Without life on land, what covers the surface is something more like weathered rock. Gravel, dust, stone that has been broken down by wind and water and chemical change, but not by roots, not by organisms, not by the patient biological work that transforms mineral into earth. The rivers that cut through this landscape carry sediment into the sea, just as rivers do today. But the sediment is different. There's no organic matter in it. The riverbeds are bare, the banks are bare, the plains beyond are bare. The colors are ochres and reds and grays and dusty browns, the colors of iron-bearing rock, of quartz, of minerals, shaped only by physics and chemistry with no life to soften them. It is beautiful in its way, a terrible ancient beauty that belongs to no human category of landscape. This is where we begin. The name that geologists have given to the particular stretch of time surrounding this moment is, on the surface, anticlimactic. They call it the boring billion. Do not be misled by the name. It was coined with a certain dry scientific humor, an acknowledgement that, from the perspective of geological drama and mass extinctions, continental collisions, explosive evolutionary radiations, the interval between approximately 1.8 billion years ago and 800 million years ago appears curiously stable. The chemistry of the ocean and atmosphere holds roughly steady. The diversity of microbial life, as best we can tell from the fossil record, does not expand dramatically. There are no sudden catastrophes, no ice ages, no great dying events, just time. Vast, patient, geochemically stable time. And yet, within that stability, the foundations of everything we are were quietly being laid. To understand the boring billion, you have to understand what came before it, approximately 2.4 billion years ago. More than a billion years before this moment, the Earth underwent one of the most radical transformations in its history. Cyanobacteria, organisms not so different in principle from those building the Stromatolites on our imagined shore, had been releasing oxygen into the ocean and atmosphere as a byproduct of photosynthesis for hundreds of millions of years. For a long time, that oxygen was mopped up almost immediately by reactions with iron dissolved in the seawater. The iron rusted. It fell to the seafloor. It formed the banded iron formations, and those ancient striped rocks of red and dark gray that miners today extract for steel, whose layered patterns are a direct record of this oxygen-producing era. The iron soaked up the oxygen again and again for hundreds of millions of years. And then, around 2.4 billion years ago, the iron ran out. Or rather, the ocean could no longer absorb the oxygen fast enough. The oxygen began to escape into the atmosphere. And for organisms that had evolved in an oxygen-free world, this was not a gift. It was a catastrophe. Oxygen is chemically reactive, biologically toxic to many microbes, a poison that burns through organic molecules. The Great Oxygenation Event, as it is called, was almost certainly one of the largest extinction events in the history of life, though we can barely detect it in the fossil record because the organisms involved were microscopic. For the survivors, and there were survivors, oxygen was the beginning of a new world. Aerobic metabolism, the ability to burn organic molecules using oxygen, is far more efficient than the older anaerobic pathways. It releases far more energy. It makes possible, eventually, complexity. But that eventual complexity is still far in the future from where we stand now. By the boring billion, by our moment 1 billion years ago, the atmosphere contains oxygen, but not in concentrations approaching today's. Current estimates suggest perhaps 1 to 4% oxygen in the atmosphere compared to the 21% of the modern world. Enough that some aerobic organisms can thrive. Not enough to build the thick oxidizing layer that would protect complex life from ultraviolet radiation on land. The ozone layer, that thin shield of oxygen molecules high in the atmosphere that blocks the most damaging solar radiation, exists, but it is thin. Thinner than today, the land surface receives a more intense wash of ultraviolet light than modern land ecosystems could survive. This is one reason among several why land remains bare. Life clings to the sea where water absorbs much of that radiation. The ocean, meanwhile, tells its own story. By the Middle Proterozoic, much of the deep ocean may have been anoxic, lacking oxygen entirely, and rich in hydrogen sulfide. The shallow waters where sunlight reaches host the cyanobacteria and their community of photosynthesizers. But below the photic zone, below the reach of light, conditions may have resembled a chemistry experiment more than a living sea. Euxinic, some scientists call it, sulfidic. The precise conditions are still debated, as they should be. The evidence is ancient and fragmentary, and honest science acknowledges the uncertainty. What is not debated is that this ocean, though alive, is alive in ways utterly foreign to us. There are no fish here, no crustaceans, no worms, possibly no cells with nuclei, though that is a matter of some scientific uncertainty. The first eukaryotes, cells with a nucleus, cells like our own, may have appeared somewhere in this time frame or somewhat earlier, but their early history is poorly preserved and poorly understood. If they exist at our moment, they are simple, few, and fragile. The ocean looks like an ocean, but it is not our ocean. And the silence, the deep, absolute silence of a world without complex life, is perhaps the strangest thing of all. On the seafloor and beneath the crust, forces move that will eventually give the world a new face. The continents of the modern world, those familiar shapes we see on every map, those boundaries we have memorized since childhood, did not always exist as they are now. They have been moving for billions of years, driven by the slow churning of the mantle beneath the crust, that sluggish conveyor of rock that carries the tectonic plates like passengers on a belt that moves at the speed of a growing fingernail. In our moment, 1 billion years ago, the continents are not arranged as you know them. Most of the world's landmass is gathered into a single supercontinent. Geologists call it Rodinia, from the Russian word for homeland or birthplace. Rodinia is not a perfect continuous mass. There may be shallow seas threading through it, interior basins and rifts and mountain ranges, but it is fundamentally one body of land, vast and mostly barren, red, brown, and gray under that hazy sky. It straddles the equatorial and mid-latitude regions, though its exact configuration is a matter of ongoing scientific investigation. The jigsaw puzzle of its pieces, cratons, the ancient stable cores of the modern continents, has been reassembled by geologists using the signatures of ancient magnetic fields frozen into the rock, the matching of geological formations across what are now different continents, the chemistry of ancient sediments. It is painstaking work, and the details remain contested. What is generally agreed is that Rodinia existed. It was real. It may have been assembled over hundreds of millions of years, reaching something like its maximum extent around 1.1 billion years ago, and it will begin to fragment somewhere around 750 million years ago, still a quarter of a billion years in our future. Standing on Rodinia's interior would be perhaps the most disorienting experience of this entire imagined journey. Imagine a landscape with no vegetation, not even a single blade of grass, not even a bacterial crust visible to the naked eye on most rock surfaces. Just stone, endless stone, mountains worn by rain and wind, valleys carved by rivers that run clear because there is nothing organic to color them, flat plains of sediment baking in the sun. The wind would sound the same as any wind, and it is only air, only pressure, only physics. but the absence of any sound from life would make it feel like wind on another planet. The rivers of Rodinia carry unusual chemistry. On a modern Earth, rivers leach organic compounds from soil and vegetation, and this chemistry is part of what makes river water the complex, life-supporting medium it is. On Rodinia, the rivers carry minerals and salts dissolved from bare rock. They deposit sediments in fans and deltas that are, in geological terms, recognizable. The physics of water and sediment do not change with or without life, but the chemistry is different, and the biology of the deltas, the life that colonizes them, is absent. Somewhere in the interior of this supercontinent there are ancient mountain belts. The Grenville orogeny, in a massive episode of mountain building associated with the assembly of Rodinia, was still relatively recent in our moment. The Grenvillian mountains may have been tall, perhaps comparable to the modern Himalayas, though by our moment they would already be in decline, eroded by hundreds of millions of years of rain and wind. Their roots are still visible in the geological record today, in the Adirondacks of New York, in the highlands of Scandinavia, in the ancient cores of continents on multiple modern land masses. They are among the oldest mountain belts we can clearly identify, and they were built in a world without animals to walk their slopes. The coast of Rodinia faces those wide, ancient oceans, and it is on those coasts, in the shallow, sunlit margins where the continent meets the sea, that the Stromatolites reach their greatest abundance. Not towering structures, not a reef in any sense we would recognize. Low mounds and ridges, some just centimeters high, some reaching perhaps a meter, a living landscape of sorts, though made by life no more complex than a colony of bacteria working in concert. There is no record of a human eye ever seeing this coast. There is no record of any eye ever seeing it. Vision had not yet evolved. The eyes that will eventually look at the natural world, the compound eyes of arthropods, the camera eyes of vertebrates, all the variations of light-sensing organ that evolution will invent over the next 500 million years, none of them exist yet. The Proterozoic shoreline was never seen by anything. We can only imagine. Beneath the surface of those ancient seas, chemistry moved in slow, patient cycles that would eventually determine the fate of every organism that has ever lived since. The Proterozoic ocean was not a dead place. It was, in fact, extraordinarily biologically active, but that activity was invisible in any way we would recognize. No schools of fish turned silver in the sunlight, no whales sounded in the deep, no coral polyps built their calcium carbonate castles on the shallower banks. The biological work of the ocean was molecular, cellular, microbial, conducted at scales we cannot see and on time scales we cannot feel. The chemistry of the seawater itself was unusual by modern standards. Dissolved iron was more abundant in some zones. Sulfate concentrations may have been lower than today. The deep ocean, as mentioned, may have been euxinic, rich in hydrogen sulfide produced by sulfate-reducing bacteria working in the anoxic depths. These are organisms that breathe, in a sense, sulfate rather than oxygen, releasing hydrogen sulfide as a waste product the way we release carbon dioxide. In the shallows, the situation was different. Here, the cyanobacteria were releasing oxygen. The oxygen reacted with iron dissolved in the water, precipitating iron oxide minerals that sank to the seafloor. This process was particularly active earlier in Earth's history. Those banded iron formations, those striped rocks of economic importance today, were mostly deposited between 2.5 and 1.8 billion years ago. By our moment, the rate of banded iron formation had slowed dramatically because the ocean had largely exhausted its supply of dissolved iron in the way that a sponge, once wrung out, cannot absorb more water until it is wetted again. The shallow seas surrounding Rodinia would have hosted the most diverse communities of life on Earth. Diverse in microbial terms. Cyanobacteria in their many forms. Other photosynthetic bacteria. Heterotrophic bacteria consuming the organic matter produced by their photosynthetic neighbors. Archaea, threading through the community, following their own ancient biochemical paths. Perhaps already the earliest eukaryotes, those cells with nuclei, those ancestors of everything we can see with the naked eye, beginning their uncertain and fragile existence. The eukaryotic story deserves a pause because it is remarkable. The eukaryotic cell, the kind of cell that makes up your body and the body of every animal, plant, and fungus on Earth, is itself the product of an ancient merger. At some point, more than 1.5 billion years ago, perhaps as early as 2 billion years ago, a bacterial cell was engulfed by a larger cell, or possibly the two fused through some other process we do not fully understand. Instead of being digested, the smallest cell survived inside the larger. Over time, it became an organelle, the mitochondrion, the power plant of the eukaryotic cell. A second merger, possibly involving a photosynthetic cyanobacterium, eventually gave rise to the chloroplast, the organelle through which plants capture sunlight. These two events, these ancient swallowing events, underlie almost all complex life. Without them, we would not exist. Without them, the history of Earth would be a history of bacteria and archaea from beginning to end. By our moment, the mitochondrion is already ancient, the eukaryotic cell is already old, but the eukaryotes themselves have not yet discovered the organizational principle that will unlock their greatest potential: multicellularity, the ability to form bodies, structures made of many cells, coordinated, differentiated, specialized for different tasks. That discovery is still in the future. For now, life is single-celled. Every organism in this ocean, as far as we can tell, consists of one cell living one life. Communities exist. The stromatolite is a community. But no organism has yet invented the trick of being many cells that function as one. The sea does not know what it is about to become. It will take another 200 or 300 million years, and it will begin not with a bang, but with a cold. Let us return to the surface for a moment, to the sky. There is something deeply strange about the sky over this Proterozoic world, and it is not only the color, though the color is different. Without the abundance of aerosols produced by vegetation, without the complex atmospheric chemistry of a biologically active land surface, the sky over the Proterozoic Earth may have had qualities we can barely model. Hazy, perhaps, from volcanic aerosols. The blue of it dependent on the same Rayleigh scattering that gives us our blue sky today, but the quality of the light subtly different, the haze of the horizon influenced by a different chemical mix. But the strangest thing about the sky is not its color. It is the speed of what moves through it. The Earth rotates faster here. This is not a metaphor or an approximation. It is measurable in ancient rock, in the layering of stromatolite structures, in the chemistry of tidal sediments that record the rhythm of lunar and solar cycles. The Earth has been slowing down since its formation, braked by the gravitational interaction with the Moon, its rotational energy gradually transferred to the Moon's orbit, pushing the Moon further away as the Earth slows down. This process, called tidal friction, has been operating for billions of years. By our moment, a day is probably somewhere between 18 and 22 hours long. Estimates vary depending on the model and the evidence used, and the honest answer is that we have a range rather than a precise figure, but shorter than today, meaningfully shorter. The consequence is a year measured in more days, not a longer year in terms of Earth's orbit around the Sun. The orbit changes much more slowly, but more rotations within that orbit. A year in the Proterozoic may have contained somewhere between 400 and 500 days, each one shorter than a modern day. The sun rises and sets faster. The quality of morning and evening light changes. The rhythm of temperature across a day is compressed. For any organism sensitive to these rhythms, and nearly all life is, this is a world calibrated differently. And above, through the night, the moon is closer. Not dramatically closer. We are not talking about a moon that fills the sky, a moon from a science fiction painting. The moon has already been receding for billions of years by this point, But a billion years of recession at a few centimeters per year accumulates to tens of thousands of kilometers. The moon of the Proterozoic is perhaps 20 or 30,000 kilometers closer than the moon we see tonight. It is a few percentage points larger in apparent diameter, but its gravitational pull on the Earth's oceans is meaningfully stronger. The tidal force drops off with the cube of the distance, so even a modest change in distance produces a noticeable difference in tidal range. The tides of the Proterozoic shoreline are larger and faster than ours. They inundate the coastal flats more rapidly, retreat more quickly, expose and cover the stromatolite beds in a cycle that differs from anything alive today. The stromatolites have grown up in these tidal rhythms, have been shaped by them, encode them in their layering. When scientists today look at ancient stromatolite structures and count the fine laminae, the individual growth layers, they are, in a sense, reading the record of ancient tidal and daily cycles. This is one of the ways we know the day was shorter and the moon was closer. Life itself recorded it. There are no seasons in the Proterozoic exactly, or rather, there are seasons, variations in solar intensity as the Earth's axial tilt causes one hemisphere to lean toward the sun and then away. But without vegetation, without animals, without any of the biological indicators we associate with seasonal change, those seasons leave only subtle chemical and sedimentary traces. The world does not go golden in autumn. It does not bloom in spring. The ice does not retreat from northern lakes. The only seasonal signal is in the chemistry of the sediment and perhaps in the growth patterns of microbial communities responding to variations in light and temperature. A year passes, and another, and a million, and a billion. This is the boring billion. We have spoken of what life was not doing, not building bodies, not walking on land, not swimming with fins, not seeing with eyes, but it is equally important to understand what life was doing. Because the microbial world of the Proterozoic was not static, it was not simple in any meaningful sense, it was quietly, invisibly revolutionary. The cyanobacteria deserve particular attention. These organisms, which still thrive today in every environment from hot springs to Antarctic ice to the open ocean, were, in the Proterozoic, the dominant photosynthesizers of a world that had no other. They fixed carbon from carbon dioxide, they released oxygen, they built stromatolites, they drove the chemistry of the shallow sea. In a world with no plants, no algae visible to the naked eye, no forests, they were the primary producers, the base of whatever food web existed, and the food web was more complex than it might appear. Bacteria consume other bacteria. Archaea extract energy from the chemical gradients produced by microbial metabolism. Viral communities circulate through microbial populations, shaping their evolution by transferring genetic material horizontally across species in ways that have no parallel in our more familiar biological world. The Proterozoic ocean, invisible though it is to us, was a seething community of interacting organisms competing and cooperating in chemical rather than physical terms. The early eukaryotes, if they existed in our moment, would have been navigating this world at a disadvantage. They were larger than bacteria, and size is expensive. A larger cell requires more energy to maintain. A larger cell moves through fluid more slowly relative to its size, but size also offers advantages. A eukaryotic cell can engulf a bacterial cell and eat it, something that bacteria cannot do in the same way. And if some of those engulfed bacteria survive and become symbionts, if the relationship becomes over time permanent, a mutualism locked into the cell's structure, then size becomes the entry point for a revolution. That revolution was already ancient by our moment. The mitochondrion had already been incorporated. But the full flowering of eukaryotic potential, the exploration of multicellularity, of differentiated cell types, of bodies, lay ahead. Somewhere in the Proterozoic ocean, in those quiet billions of years, the ancestors of animals were alive. Small, single-celled, unremarkable. They would not have looked like animals. They would have looked like the other protists Around them, a cell, a nucleus, a few organelles, a membrane, a life. But their particular lineage of genes, their particular metabolic architecture, their particular way of handling certain proteins involved in cell adhesion and signaling, these were the deep blueprint from which, given enough time and the right circumstances, bodies would eventually be built. The fossil record for this period is frustratingly sparse. Microbial cells do not fossilize easily. When they do, they often preserve only their outer walls, a shape, a hint of structure, with no chemical or genetic information surviving across 1 billion years of geological transformation. The Proterozoic fossil record is a whisper, not a shout. Fragments of ancient microbial communities, chemical signatures in ancient sediments, isotopic ratios that record the action of photosynthesis and respiration, and sulfate reduction. These are the clues we have. They are enough to sketch an outline. They are not enough to fill it in completely. And so we sit with uncertainty, honest uncertainty. We know what was probably there. We know what probably was not. We can only imagine the details, and that imagination is constrained by what we know of chemistry, of biology, of the physics of the ancient earth. The microbial world did not know it was about to change. It had been stable roughly for hundreds of millions of years. The boring billion had been, for its inhabitants, not boring at all. It was the entire world, and they were its masters. And then slowly something shifted. The stability of the boring billion did not last forever. It could not last forever. The universe does not permit permanent stasis, and the Earth, a restless planet, driven by the heat of its own formation and the decay of radioactive elements deep in its interior, was always accumulating change, even when that change was invisible. Around 800 million years ago, still in the future from our standing point of 1 billion years but coming, Rodinia will begin to break apart. The supercontinent will fragment along ancient lines of weakness, rifts opening in the lithosphere as plumes of hot mantle rock rise beneath the crust and split it. The pieces will drift away from each other, opening new ocean basins, rearranging the circulation of those ancient seas, changing the pattern of rainfall and weathering on the land. That breakup will have consequences. The increased length of coastline as continents fragment means more shallow shelf environments. And shallow shelf environments are where life is richest, where nutrients are most concentrated, where sunlight can reach the seafloor. The weathering of newly exposed rock as rifting opens new mountain belts, draws down carbon dioxide from the atmosphere, cooling the climate. Carbon dioxide is a greenhouse gas. Removing it cools the earth. This cooling will contribute over millions of years to something extraordinary and terrifying: snowball Earth. The cryogenian glaciations, the extreme ice ages that will follow the breakup of Rodinia, centered around 700 million years ago, are among the most dramatic events in the history of the planet. Evidence suggests that glaciers may have extended to the equator, that the entire ocean surface may have been covered with ice, that the Earth may have been a frozen ball for millions of years before volcanic carbon dioxide, slowly accumulating beneath the ice, warmed the planet enough to melt it. Whether these snowball Earth episodes were total, a fully frozen planet, or partial, leaving some open water in the tropic, is still debated. The evidence points toward extreme glaciation, but the details remain uncertain. What is clear is that the cryogenian was a time of environmental stress unlike anything in the preceding boring billion, and stress drives evolution. The organisms that survived the snowball Earth episode, if they were as extreme as some models suggest, were those with the greatest metabolic flexibility, the greatest ability to persist under radical environmental change. They were the organisms that, given the right circumstances, might find new ways of being. In the immediate aftermath of the cryogenian glaciations, something changes in the fossil record. The diversity of eukaryotic organism, those complex cells with nuclei, begins to increase. Evidence for multicellularity, tentative and debated, begins to appear. The Ediacaran period that follows the Cryogenian will eventually produce the first macroscopic organisms visible to the naked eye, the Ediacaran fauna, soft-bodied creatures of uncertain relationship to anything alive today, mat-dwelling organisms that covered the seafloor in forms that had no predecessors and left few descendants. But all of that is still in the future. For now, in our moment of 1 billion years ago, the boring billion continues. The stromatolites grow, the cyanobacteria photosynthesize, the oxygen accumulates molecule by molecule. The day is short, the moon is near, the land is bare, and deep in the chemistry of the ocean, in the genes of organisms too small to see, the blueprint of everything that will follow is being written in a language we are only beginning to translate. How do we know any of this? It is a fair question, and it deserves a direct answer. We are speaking about a world that existed 1 billion years ago, a world that left no written records, no artifacts, no structures built by minds that could intend them. The evidence is geological, chemical, and biological, preserved in ancient rock, in the chemistry of minerals, in the microscopic structures of fossilized cells. The banded iron formations are among our most important witnesses. Those layered rocks of red and dark gray that exist on every continent, in the Pilbara of Western Australia, in the Anamiki Basin of the Great Lakes region, in the iron ranges of Brazil and Russia and South Africa, were deposited when dissolved iron in ancient seawater reacted with oxygen produced by photosynthesis. Their layering reflects cycles of oxygen production and iron deposition. Their chemistry records the redox state of the ancient ocean. They are a library written in mineral, and scientists have been reading them for more than a century. The stromatolites themselves are archives. Their fine laminae record tidal and daily cycles. Their chemistry records the isotopic signatures of ancient carbon, oxygen, and sulfur cycling. Some ancient stromatolites preserve the shapes of the cells that built them. The ghostly outlines of cyanobacterial filaments or colloidal cells, mineralized by silica or carbonate over hundreds of millions of years, still visible under a microscope. When a scientist holds a thin section of ancient chert, ancient silicified rock, up to a microscope light and sees the cellular structures inside, they are looking at life that lived 1 billion years before their own birth. The isotopic record is perhaps the most subtle and most powerful tool. Carbon, sulfur, oxygen, strontium. All of these elements have multiple isotopes, slightly different versions of the same atom whose relative abundance in ancient rocks reflects the chemistry of the environment in which those rocks formed. Photosynthesis preferentially incorporates lighter carbon isotopes, leaving a signature in organic matter. Sulfate-reducing bacteria fractionate sulfur isotopes. The isotopic ratios preserved in ancient sedimentary rocks allow scientists to reconstruct the chemistry of ancient oceans and atmospheres with remarkable precision, or, more accurately, with a precision that is remarkable given the age of the material, while still carrying real uncertainties that honest scientists acknowledge. Paleomagnetics, the study of ancient magnetic fields recorded in rocks, is how we reconstruct the positions of ancient continents, including Rodinia. When molten rock cools and solidifies, magnetic minerals within it align with the Earth's magnetic field, recording the direction and intensity of that field at the moment of solidification. Over time, as the tectonic plate carrying that rock moves across the globe, the recorded magnetic direction comes to point in a different direction from the current pole. By measuring the discrepancy, scientists can calculate where on the globe the rock was when it formed. Do this for many rocks from many continents, and you can begin to reconstruct the ancient arrangement of the plates. It is detective work, patient, meticulous, subject to revision as new evidence emerges. The reconstruction of Rodinia has changed many times over the past few decades and will change again. The history we are telling here is the best current picture. A picture assembled by many scientists across many years, always held provisionally, always open to revision. This is not a weakness. This is the nature of knowledge about a vanished world. The rocks do not lie, but they are not always legible. We read them as carefully as we can, acknowledge what we do not know, and continue. There is a feeling that sometimes overtakes the people who study the deep past. A geologist standing in a canyon wall reading strata laid down over millions of years, a paleontologist holding in their hands the mineralized remains of an organism that lived before eyes existed, an astrobiologist modeling the chemistry of an ancient ocean in a computer, watching the simulation unfold over billions of simulated years. The feeling is not quite awe, though it contains awe. It is not quite humility, though it contains that too. It is something closer to a shift in scale, a sudden, vertiginous sense of how brief human existence is, how recent, how small in the temporal landscape of a planet that has been changing for 4.5 billion years and will go on changing for billions more. The boring billion is boring only from the perspective of geological drama. From the perspective of any individual life form within it, if such a thing as perspective could be attributed to a cyanobacterium, each moment was everything. Each pulse of sunlight was everything, each chemical gradient was everything. Life does not experience geological time, life experiences only the present moment, the immediate chemistry, the immediate pressure of survival and reproduction, and in that immediate present repeated trillions of times across 1 billion years, the work of evolution continued. Slow, undirected, patient in the way that only mindless processes can be patient. Not patient because it waited, but patient because it simply continued, one chemical reaction at a time, one cell division at a time, one mutation at a time. We sometimes speak of geological time as if it were experience, as if the earth waited impatiently or patiently for the next chapter to begin. It did not. Time is experienced only by minds, and the Proterozoic had no minds. What happened in the boring billion happened without anyone watching, without anyone counting, without anyone aware that a story was unfolding, and yet the story was unfolding. One cell division at a time, one oxygen molecule at a time, one crystal of iron oxide settling to the seafloor at a time. The organisms of the Proterozoic ocean did not know they were laying the foundations of everything. They did not know about the Cambrian explosion still hundreds of millions of years ahead, when animal body plans would diversify in what seems geologically like a burst of creativity. They did not know about the Ordovician radiation, or the colonization of land by plants and animals, or the rise of forests, or the evolution of flight, or the emergence of consciousness. They knew nothing. They simply lived in the only way available to them in a world that was the entirety of existence. We are the ones who look back. We are the ones who try to see the whole arc, who try to trace the thread from those ancient microbial communities to ourselves. And when we succeed even partially, even tentatively, the result is something that feels less like scientific knowledge and more like a kind of time travel, a genuine connection across 1 billion years to something that was alive, that breathed the chemistry of an alien world that persisted. The Stromatolites in the shallow seas of Rodinia were alive. That is the most astonishing thing. In a world so different from our own that we could not breathe its air or drink its water, in a world with no land life and no animals and no eyes to see it, there was life, patient, persistent, planet-altering life. Life that we are still connected to through the unbroken chain of reproduction that links every living thing on Earth to every other. You exist because those cyanobacteria released oxygen. You exist because some ancient cell 1 billion years ago or more merged with another and became the ancestor of your own cells. The mitochondria in your cells right now are the descendants of those ancient bacterial symbionts. Your own body is, in some sense, an ecosystem, a community of cells descended from ancient single-celled organisms that made an accommodation with each other so deep that we no longer think of ourselves as many, but as one. Think of the patience required, not patience in any conscious sense, but the patience embedded in the structure of the universe itself. the patience of a process that runs for 1 billion years without any external pressure to hurry, without any reward for speed, without any direction other than the blind gradient of chemistry seeking equilibrium. It is a patience we cannot emulate and can barely comprehend. We are creatures of decades, of years, of days. 1 billion years defeats any attempt at personal analogy, and yet here we are on the far side of it, having benefited from every moment of it. The boring billion was not boring. It was the crucible, the long patience in which the prerequisites for everything were assembled, one molecule at a time. Look at the ocean, not at its surface. The surface can mislead you. Look into it, into the water column, into the shallow shelf zones where the light penetrates and the chemistry is most active. And now consider a question that seems simple, but is not. What color is this ocean? The modern ocean is many colors depending on where you look. In the open, deep ocean, far from land and sediment, the water appears a deep, pure blue, almost indigo. The color of pure water absorbing the longer wavelengths of light and scattering the shorter blue ones back to the surface. In coastal areas, the presence of phytoplankton, those microscopic photosynthetic organisms that color so much of the modern ocean, can shift the color toward greens and blue-greens. In river mouths and estuaries, sediment turns the water brown or gray. The Proterozoic ocean did not look like this. The shallow shelf areas near Rodinia were populated by cyanobacteria, and where cyanobacteria are abundant, they impart a distinctive color, blue-green, but often tinged with other pigments. Different groups of cyanobacteria contain different accessory pigments, phycocyanin, phycoerythrin, that can shift their color toward red or orange or yellow, depending on which wavelengths of light are available and which pigments have been selected for. Phycoerythrin in particular is found in cyanobacteria that live at greater depths, where red light has been absorbed and the remaining light is primarily blue-green. Phycoerythrin absorbs this light efficiently and can make cells appear red or orange. If the deep ocean was rich in hydrogen sulfide, as some models of the Proterozoic suggest, then sulfur oxidizing bacteria, which form white or yellow mats, may also have been present near the chemicline, the boundary between oxygenated and anoxic water. The ocean may have had layers of visible color, zones of chemical transition that created bands of microorganisms, each adapted to the conditions of their particular depth. In the open, deep ocean, far from the continental margins, the water may have appeared much as it does today. Deep blue, its color determined primarily by the physics of pure water rather than by biology. The cyanobacteria of the open ocean were too sparse to significantly color the water visible from above. But the shallow seas, the shelves, the restricted basins, the inland seas that might have threaded through the interior of Rodinia, these may have had colors we would find arresting. Green-blue near the surface in areas of dense cyanobacterial growth, perhaps a rust-red tinge where iron was precipitating out of solution, perhaps milky white in zones of calcium carbonate precipitation, because cyanobacterial photosynthesis can drive the precipitation of calcite in supersaturated waters, creating whitings, those sudden blooms of suspended calcium carbonate that still occur today in the Bahamas and the Black Sea. We cannot know for certain. The colors of an ancient ocean are not preserved in rock, but the chemistry is preserved, and the chemistry constrains the color, and we can make reasonable inferences about what a Proterozoic sailor, had there been any such thing, might have seen looking over the rail of a ship that could not exist. What they would have seen almost certainly was not the clear blue of a coral sea. It was an older color, a denser color, a color that spoke of chemistry at work, iron and sulfur and oxygen in slow negotiation, life threading its way through the gradients, building and consuming and building again. The ancient ocean was alive, it just did not look like ours. We use the word weather so casually that we rarely pause to notice what a complex biological and physical system it describes. The weather we experience, rain, wind, temperature, humidity, is shaped not just by physics but by the living world. The trees and plants that cover much of the modern land surface release enormous quantities of water vapor into the atmosphere, feeding the hydrological cycle. They release volatile organic compounds that serve as nuclei for cloud formation. They alter the albedo of the surface and its reflectivity compared to bare rock. They pump carbon dioxide out of the atmosphere and store carbon in their bodies. They stabilize the soil against erosion, and their roots break down rock in ways that release nutrients and alter the chemistry of the runoff that feeds the rivers. In the Proterozoic, none of this biological modulation of the weather system existed on land. The land surface was bare rock and sediment. The hydrological cycle operated by the same physical laws it does today. Water evaporates from the ocean, rises into the atmosphere, condenses into clouds, falls as rain, flows back to the sea. But without vegetation to hold and release moisture on land, without organic matter in the soil to absorb and slowly release it, the cycle may have been more extreme. Rain that fell on bare rock ran off immediately into rivers and the sea, rather than being retained in the soil and transpired slowly back into the atmosphere over days and weeks. The continental interiors may have been drier than they would be today with the same geography, because the mechanism of vegetation-driven moisture recycling, where trees effectively pass ocean moisture inland by transpiring water that falls as rain further and further from the coast, did not exist. This means that the deep interior of Rodinia, far from the ocean, sheltered from rain by the mountain ranges at the margins, may have been extraordinarily dry. An arid interior of bare rock subjected to extreme temperature swings between day and night with no moderating influence of vegetation or soil. The temperature of a bare rock surface in full sunlight can reach 60 or 70 degrees Celsius. The same surface at night can plunge below freezing. Without soil and vegetation to buffer these extremes, the Proterozoic continental interior may have experienced daily temperature swings that would challenge any modern ecosystem. The rivers that drained this landscape were unusual. Modern rivers carry a load that is partly mineral sediment and partly organic matter. Leaves, wood fragments, dissolved organic carbon from decomposing vegetation. Proterozoic rivers carried only mineral sediment. Their chemistry was different, and their behavior was different. Without the cohesion provided by plant roots and organic matter in riverbanks, Proterozoic rivers may have been more likely to braid, to spread into wide, shallow networks of shifting channels, rather than to cut the deep, single-channel valleys that we associate with vegetated landscapes. The great canyons and river valleys of the modern world owe much of their character to the presence of life on land. Without it, the rivers would have wandered differently. Storms would have been different too. Without the influence of land vegetation on the hydrological cycle, storm patterns over the continents may have differed from today. Lightning, produced by electrical discharge in storm clouds, would still have occurred, still have struck the bare rock, and possibly ignited whatever organic matter existed on the surface, though there was little enough to burn. The physics of the atmosphere is largely independent of biology at the scales that determine weather, but the finer details, the distribution of moisture, the formation of clouds, the chemistry of rainfall, these were shaped by a land without life, and they shaped a land without life in return. It is a feedback loop we have broken so completely with our civilization that we tend not to notice it was ever there. But the bare rock and sediment world of the Proterozoic is as close to the original planet as we can imagine. Let us think carefully about who was alive in this moment 1 billion years ago. Not what was absent. That list is long and obvious. Who was present? The bacteria, numerically, metabolically, ecologically dominant. The cyanobacteria we have discussed at length. But also the sulfate-reducing bacteria of the anoxic deep ocean, the iron-oxidizing bacteria of the chemocline, the methanogens, archaea that produce methane as a byproduct of their metabolism, working in anoxic sediments, generating that faint sulfurous sweet smell that still rises from oxygen-depleted mud today. The purple sulfur bacteria and green sulfur bacteria using hydrogen sulfide instead of water as their electron donor for photosynthesis, releasing sulfur rather than oxygen. Each of these lineages was ancient by this point, billion-year-old traditions of metabolism that had been refined over geological time into extraordinary efficiency. The archaea, often invisible in popular accounts of early Earth, often overshadowed by their better-known bacterial cousins, but no less ancient, no less important. The archaea includes some of the most metabolically diverse organisms ever to have lived: the methanogens, the thermophiles that thrive in hot springs at temperatures that would cook any familiar organism, the halophiles that live in salt concentrations 10 times that of seawater, the acidophiles that thrive in conditions of extreme acidity. By the Proterozoic, all of these traditions were already ancient. The archaea had been practicing these extremophile lifestyles for longer than animals have existed. And then there are the eukaryotes. The question of when exactly eukaryotes evolved is a matter of ongoing scientific discussion. The molecular clock, the technique of using the rate of mutation in genes to estimate the timing of evolutionary divergences, suggests that eukaryotes may have originated somewhere between 1.5 and 2 billion years ago. The fossil record offers a few tantalizing candidates for early eukaryotic fossils, but interpreting microfossils is notoriously difficult. By 1 billion years ago, it is likely that eukaryotes existed, though their diversity and abundance relative to bacteria and archaea is unknown. What we can say with reasonable confidence is that by approximately 1.2 billion years ago, sexually reproducing eukaryotes existed. Bangiomorpha pubescens, a fossil red alga found in Arctic Canada, was described from rocks dating to roughly this age, and it shows evidence of sexual reproduction in its cellular structure. Sexual reproduction is a eukaryotic invention and a profound one. Without sex, genetic variation accumulates only through mutation, a slow, one-at-a-time process. With sex, an organism can combine its genes with those of another individual, producing offspring with entirely novel combinations that natural selection can then test. This accelerates evolution enormously, and it was a development whose consequences would not become fully apparent for hundreds of millions of years, but which began in the Proterozoic. Red algae, rhodophytes, are among the oldest lineages of multicellular eukaryotes. They have a fossil record stretching back more than 1 billion years in some interpretations. They still exist today, forming the pink and red seaweeds of rocky shores worldwide, incorporated into the walls of every Japanese restaurant as nori, contributing their characteristic colors to coral reef ecosystems. When you handle a piece of seaweed, you are handling the descendant of one of the oldest multicellular lineages on Earth, a lineage that was already ancient by our Proterozoic moment. The green algae, chlorophytes, were also probably present in some form by 1 billion years ago. And the green algae matter in a way that is not immediately obvious, because the green algae are the ancestors of all land plants. Every tree, every flower, every blade of grass, every fern and moss and liverwort that has ever grown on the continents of Earth, all of them descended from green algae that made the transition to land beginning around 500 million years ago. The first organisms to colonize bare Proterozoic rock, and not yet, but eventually, were the ancestors of everything green. They were in the ocean 1 billion years ago, simple, green, single-celled, or forming simple filaments. They had no idea they were ancestors. They were simply alive, doing what they had always done. There is a particular kind of beauty in the way that living processes leave permanent records in stone. Consider the banded iron formations again, those ancient striped rocks that exist on every continent, whose red and dark gray bands stretch for kilometers, that feed the steel industry of the modern world. These rocks were made by life, not by any individual organism, not by any intentional act, but by the accumulated chemistry of billions of organisms releasing oxygen as a byproduct of their own survival. That oxygen reacted with iron. The iron precipitated. It fell to the seafloor. It became rock. And that rock, after billions of years of burial and metamorphism and tectonic activity, is now exposed at the surface, where we can find it and study it and recognize it for what it is, a monument to ancient photosynthesis. The largest banded iron formations were deposited before our moment, and most of them are more than 1.8 billion years old, but smaller deposits continue to form in the Proterozoic in restricted basins and anoxic environments where iron could still accumulate. They are scattered across the geological record like bookmarks in a very long story. The stromatolites, too, become stone, or rather, they create structures that can be lithified, turned to rock by the gradual replacement of organic material with mineral. Fossil stromatolites are found on every continent in rocks of vastly different ages. The oldest are more than 5 billion years old, found in the Pilbara region of Western Australia. Those of the Proterozoic, our era, are particularly abundant and diverse. They come in cones and domes and sheets and branching forms, each variety reflecting the particular water energy and chemistry and tidal regime in which it grew. Reading the variations in ancient stromatolite morphology is, in a sense, reading ancient geography. The shape of ancient coastlines and shallow seas, preserved in stone. Phosphorite deposits, rocks rich in phosphate, also record ancient microbial activity. Phosphorus is a nutrient essential for life, and in certain marine environments where microbial activity concentrates it, it precipitates as minerals that can form large deposits. Ancient phosphorites are found worldwide, and they record episodes of intense biological activity in ancient seas. The Proterozoic rock record is, in fact, surprisingly rich, surprisingly accessible to the geological eye. The oldest rocks on Earth date to more than 4 billion years ago, but they are rare, highly metamorphosed, and extremely difficult to interpret. The Proterozoic rock record, while old, is less heavily altered. It preserves sedimentary structures, fossil communities, chemical signatures that can be analyzed with modern laboratory techniques. The Great Lakes region of North America, the Baltic Shield of Scandinavia, the Kapval Craton of southern Africa, the Pilbara and Yilgarn Cratons of Australia. These are among the places where the Proterozoic past is most accessible, most studied, most legible. Each outcrop of ancient rock is a window. Hold a hand lens over an ancient chert and look for the dark, irregular masses that represent fossil microbial mats. The Gunflint Formation of Ontario, dating to roughly 1.9 billion years ago, contains some of the best preserved microbial fossils in the world, a community of cyanobacteria and other microorganisms caught in silica, frozen at a moment that was already ancient when the dinosaurs were young. This is the privilege of geology. The past is not entirely gone. It is here, in the rock beneath our feet, in the mineral records of ancient chemistry, in the microscopic structures of fossil cells. We can go back, not in time, but in understanding. We can reconstruct imperfectly but genuinely a world that no conscious being ever saw. There is a philosophical dimension to deep time that I want to sit with for a moment before we move toward the close. We are accustomed as human beings to thinking of time as something that flows. It moves, we say, forward, always forward, carrying us with it. We feel this flow in the rhythms of our bodies, the heartbeat, the breath, the cycle of sleeping and waking. We feel it in the seasons, in the aging of our bodies, in the accumulation of memory. Time feels continuous and it feels personal. Our time, the time we are living through, connected to a past we remember and a future we can imagine. Deep time is not like this. Deep time is not a feeling. It is a quantity so large that it defeats imagination and can only be apprehended through analogy or mathematics. 1 billion years. If each second represented 1 year, it would take more than 31 years to count to 1 billion. If each step were 1 year, it would take from here to the moon and back many times over. The numbers are too large to feel. And yet the events that happened in that time were real. They had causes and effects. The oxygen released by cyanobacteria 1 billion years ago was the same oxygen that your mitochondria are using right now. The carbon deposited in ancient organic matter during the Proterozoic is the carbon you exhale as carbon dioxide when you breathe. The connections are real, even when the time is incomprehensible. There is something consoling in this and something vertiginous. The consoling part is this: we are not separate from deep time. We are made of it. Every atom in our bodies has been through multiple stars, multiple planetary systems, multiple geological cycles. The calcium in our bones was forged in the nuclear furnaces of ancient stars that exploded before the solar system existed. The iron in our blood is star iron. The carbon that forms the backbone of our organic molecules has been part of countless living organisms across billions of years of Earth's history. We are not observers of deep time. We are participants in it. The vertiginous part is this: 1 billion years from now, something will exist where the Earth is, and it will be as different from us as we are from the cyanobacteria of the Proterozoic. Or perhaps nothing will exist. The sun is slowly brightening. In 1 billion years or so, it will have brightened enough to begin driving a runaway greenhouse effect that will eventually render Earth uninhabitable as its oceans evaporate and its atmosphere thickens with water vapor. Before that process is complete, complex life may no longer be possible on the surface. The long story that began with those Proterozoic microbes will come to an end, and for this planet in this solar system, through this particular unfolding of physics. But the atoms will continue. The carbon and hydrogen and oxygen and nitrogen and all the rest will disperse back into the galaxy, carried on the solar wind and on the gravitational currents of the Milky Way. Some of those atoms may eventually be incorporated into new planets forming around new stars. And on some of those planets, perhaps chemistry will once again organize itself into the astonishing pattern we call life. We can only imagine. Let us return one final time to the ocean. It is late in our imagined visit. The sun is lower in the sky. The short day is nearly done. And in the shallow waters before us, the Stromatolites continue their ancient work. The same work they have been doing for 3 billion years by this point, the same work they will continue for hundreds of millions of years after this moment. Consider what they have accomplished. In the beginning, more than 4 billion years ago, when the Earth was young, the atmosphere of this planet was almost entirely without oxygen. The oxygen that exists today, every molecule of it, was produced by life. Not by geological processes, not by chemistry without biology, but by living organisms, by photosynthesis, by the patient biochemical machinery that takes carbon dioxide and water and sunlight and produces organic matter and oxygen over and over and over again across geological time. The cyanobacteria did not set out to change the atmosphere. They set out only to survive, to grow, to reproduce, as every living thing does. But the aggregate effect of trillions of organisms doing this across billions of years was an atmospheric transformation without parallel in the inner solar system. Mars and Venus, our nearest neighbors, have atmospheres dominated by carbon dioxide. Earth's atmosphere today is 21% oxygen, a concentration maintained by the continued work of photosynthesis, cycling carbon into organic matter, and releasing oxygen in its place. Without continuous photosynthesis, the oxygen in Earth's atmosphere would be consumed by geological oxidation within a few million years. The Stromatolites made the world in which complex life eventually became possible. They did not intend to. They did not know they were doing it, but they did it. And there is something in that thought worth sitting with. The most consequential act in the history of Earth's biosphere was performed by organisms without minds, without purpose, without any sense of the significance of what they were doing. They were simply living. The significance was retrospective, visible only from the far side of 1 billion years. Perhaps all significance is retrospective in this way. Perhaps what matters only becomes apparent when enough time has passed to see the pattern. The tide is rising now. It covers the lowest stromatolite beds with water that catches the last light of the Proterozoic day. The short night is beginning. The stars will be different from ours. The constellations do not exist yet in their familiar forms, because the stars that make them up have been moving for 1 billion years since this moment, and the view from Earth has changed. The familiar patterns, Orion, the Big Dipper, Cassiopeia, are not there. The individual stars are the same but arranged differently, scattered across a sky that belongs to no human myth or story. And the moon rising now is close, brighter perhaps, with its greater apparent size, or perhaps not, since apparent brightness depends on the square of the angular diameter, and the difference is not dramatic, but different, measurably, meaningfully different. The sky above the Proterozoic Earth at night is the same universe we live in. The stars are there, the galaxies are there, though we cannot see most of them. The great cosmic structures, the filaments and voids and galaxy clusters that make up the large-scale architecture of the universe are all there, exactly as they will be in 1 billion years, because the universe is old enough that its large-scale structure does not change significantly over a mere billion years of cosmic time. We are not alone in the universe. We are alone, and nearly so, in this particular corner of it, this particular shore, this particular moment, and it is enough. There is another dimension to the Proterozoic world that we have not yet entered, the dimension beneath the surface. The deep interior of the Earth in this moment 1 billion years ago was doing exactly what it does today, but with more heat. The Earth formed 4.5 billion years ago from the collision and accretion of smaller planetesimals, and that collision released an enormous amount of heat. Additionally, the decay of radioactive elements, uranium, thorium, potassium, 40, generates heat continuously within the interior. In the early Earth, this radiogenic heat was far greater than it is today, because the radioactive elements had not yet decayed to their current levels. By 1 billion years ago, the heat flow from the interior had declined substantially from its primordial peak, but it was still higher than it is now. The mantle convected more vigorously, the tectonic plates moved faster, the mid-ocean ridges produced new oceanic crust at higher rates. More volcanism means more carbon dioxide delivered to the atmosphere. The carbon dioxide balance of the ancient Earth, the long-term carbon cycle that regulates climate over millions of years, was maintained by the interplay between volcanic outgassing and the weathering of silicate rocks. Carbon dioxide dissolves in rainwater, forming carbonic acid. That acid weathers silicate minerals, releasing calcium and magnesium ions, and dissolve carbonate into rivers and eventually into the sea. In the sea, organisms use calcium and carbonate to build shells. But in the Proterozoic, there were no shell-building organisms. The calcium carbonate instead precipitated directly from supersaturated seawater, forming limestone and dolostone on the seafloor. This process, the weathering of silicate rock, the transport of calcium and carbonate to the sea, the precipitation of carbonate minerals on the seafloor, constituted the primary means by which carbon was removed from the atmosphere over geological time in the Proterozoic. It was slower and less efficient than the biological carbonate precipitation that would come later when animals evolved shells and reefs, but it was sufficient. The volcanic outgassing and the silicate weathering were in balance, roughly during the boring billion. That balance maintained the climate in a moderate range, warm perhaps, but not extremely so, cool at times, but not glacial. The equilibrium was real, even if it was maintained by geological processes operating on time scales of millions of years, and somewhere in the deep ocean, in the dark and pressure of the abyssal plain, the hydrothermal vents poured their chemical wealth into the water. Not just heat, minerals, iron, manganese, sulfide, silica. The hydrothermal fluid that pours from these vents is the product of seawater, circulating through the hot rock of the oceanic crust, reacting with it at high temperature and pressure, emerging transformed. It is a geological chemistry experiment that has been running continuously for billions of years, and its products shape the chemistry of the ocean as surely as the input of rivers from the continents. The vent communities of the Proterozoic were alive. We do not have direct fossil evidence for them. The hot, chemically reactive environment of hydrothermal vents is not conducive to the preservation of microbial fossils. But the metabolic pathways used by vent chemolithotrophs are ancient, and there is every reason to believe they were present. Life had been exploiting hydrothermal energy since before the fossil record begins. In the darkness of the Proterozoic deep ocean, far below any sunlight, far below even the faint glow of bioluminescence that would eventually evolve in animals, there was life, chemical life, invisible life, life that had never needed the sun, that drew its energy from the heat and chemistry of the planet itself. It is a reminder that life, once it finds a foothold, does not restrict itself. It goes wherever the chemistry permits. It existed in the sunlit shallow seas and in the lightless deep. It existed on bare rock surfaces and in the anoxic depths. It existed in all probability wherever liquid water and chemical energy coincided, a combination that the Earth, with its geological heat and its ocean, provided in abundance. The planet was more alive in this sense than the bare rock surface suggested. There is a discipline in honest science that sometimes makes it feel less satisfying than we might wish. The discipline of acknowledging what we do not know. We have spoken throughout this journey as though we have a clear and detailed picture of the Proterozoic world. In truth, our picture has significant gaps, some of them substantial. The geological record for this period is better than for earlier eras, but it is still fragmentary. The evidence we have is indirect. Chemical signatures, mineral structures, microscopic fossils, paleomagnetic data, and interpreting it requires assumptions and models that are continually being revised. We do not know with precision how much oxygen was in the atmosphere a billion years ago. Current estimates range from perhaps 1% to perhaps 4 or 5%, a wide range with significant implications for the habitability of different environments. The details of ocean chemistry, the distribution of anoxic and euxinic zones, the concentrations of various dissolved elements, the precise redox state of different ocean basins are still actively debated among researchers. We do not know the precise configuration of Rodinia. The reconstruction of ancient supercontinents is an ongoing scientific project, and different research groups produce different models, sometimes differing significantly in the position of individual cratons. The details matter for understanding climate patterns, ocean circulation, and the distribution of life, and they remain uncertain. We do not know when eukaryotes evolved with precision. The molecular clock gives us a range, the fossil record gives us a minimum date. But the range is wide and the minimum date is not the same as the actual date of origin. The history of early eukaryotic diversity is one of the most active research areas in paleobiology, and the picture is changing rapidly as new fossil finds are made and new analytical techniques are applied to ancient rocks. We do not know what the first multicellular eukaryotes looked like, or exactly when they appeared. The Bangiomorpha fossils from the Canadian Arctic suggest sexual reproduction in red algae at approximately 1.5 billion years ago, which is close to our moment of 1 billion years. But whether multicellularity had already evolved by 1 billion years ago, and in which lineages, is uncertain. We can only imagine many of the details we have described in this narrative. The color of the ocean, the quality of the light, the smell of the air, these are informed inferences, not observations. The inference is constrained by what we know of chemistry and physics, but there is a gap between the constraint and the certainty. This is not a weakness of science. It is the nature of investigating a world so ancient that direct observation is impossible. The appropriate response is not to fill the gaps with false certainty, nor to abandon the inquiry because certainty is unavailable. The appropriate response is to reason as carefully as possible from the evidence we have, to acknowledge the limits of that evidence, and to hold our conclusions with the right degree of confidence. Firm, where the evidence is strong; tentative, where it is weak; open to revision, where new evidence might change the picture. The Proterozoic world we have described is the best current understanding. It is the picture that emerges from decades of research by hundreds of scientists, each contributing a piece of evidence, each revising and refining the model. It is not the final picture. No picture of deep time is ever final. And there's something right about that. A world so ancient should retain its mystery. A shore that no eye ever saw should resist our attempts to reconstruct it perfectly. The honest position is, we know this much, and this much is extraordinary, and there is more we do not know. The Proterozoic ocean held secrets. It still does. Some of those secrets are encoded in rock that has not yet been found or not yet analyzed with the right techniques. Future scientists will read things in that rock that we cannot yet read. They will revise this picture. They will find organisms we did not know existed, or geochemical evidence for processes we did not imagine, or tectonic reconstructions that shift the position of Rodinia's pieces. And each revision will bring us closer, not to a final answer because there may be no final answer, but to a more complete understanding of a world that was real, that existed, that mattered. The cyanobacteria on that ancient shore produce the oxygen you are breathing right now. The continental rocks of Rodinia are the foundations on which modern continents rest. The chemistry of that Proterozoic ocean shaped the evolution of life in ways we are still tracing. The boring billion was not boring. It was the foundation. And we are, in the most literal, physical sense, built upon it. The story of the Proterozoic is the story of a world approaching a threshold. For hundreds of millions of years, the long patience of the boring billion, the Earth was stable. The chemistry of the ocean was roughly constant. The diversity of life was roughly constant. The arrangement of the continents was slowly changing, but the changes were gradual and the disruptions were modest. It was a world in a kind of deep equilibrium. Not static, because nothing geological is static, but slow, constrained, cycling through patterns that repeated across geological time without decisive change in any direction. And then the equilibrium broke. The breakup of Rodinia, the rifting of that great supercontinent along its ancient seams, the opening of new ocean basins, the flooding of continental margins with shallow seas, the exposure of new rock to chemical weathering altered the carbon cycle. Carbon dioxide was drawn from the atmosphere by the weathering of silicate rocks, a process in which rainwater, naturally slightly acidic because it dissolves carbon dioxide from the atmosphere to form carbonic acid, reacts with rock minerals, incorporating the carbon into dissolved form that eventually reaches the sea, where it can be precipitated as carbonate sediment. More rock exposure means more weathering. More weathering means more carbon drawn from the atmosphere. Less carbon dioxide means a weaker greenhouse effect. A weaker greenhouse effect means a cooler climate. A cooler climate eventually means ice. And the ice of the Cryogenian, the glaciations that will follow the boring billion, was unlike anything since. Ice sheets advanced to the tropics. The albedo of the ice reflected more sunlight back into space, amplifying the cooling. And if, as some models suggest, the ocean surface froze globally, then photosynthesis would have nearly stopped, the carbon cycle would have essentially halted, and only the steady outgassing of carbon dioxide from volcanoes would have slowly, over millions of years, rebuilt the greenhouse effect enough to melt the ice. The aftermath of such extreme glaciations would have been extreme in its own right. A superheated world with vast carbonate deposits and a pulse of oxygen, and in the aftermath of that pulse, a biological diversification. But I am getting ahead of ourselves. For now, in our moment 1 billion years ago, the threshold is not yet crossed. The boring billion continues. The stromatolites grow. The cyanobacteria photosynthesize. The oxygen accumulates in the atmosphere, molecule by molecule, too slowly for any individual organism to perceive, but fast enough across geological time to transform the world. The first animals, if we can call them that, those early multicellular organisms, those early experiments in differentiated cell types and coordinated function, have not yet appeared. They will appear eventually in the fossil record of the Ediacaran after the cryogenian ice has melted. They will be soft-bodied, mat-dwelling, enigmatic. They will not have eyes or mouths in any sense we would recognize. They will be the first rough drafts of what we are. But in the present moment, our moment, there are only the microbes. The patient, unobserved, metabolically extraordinary microbes going about the business of life in their ocean, on their planet, under their nearer moon. If you could stand on that shore and wait long enough, if you could compress 1 billion years into a single sleepless night, you would watch the tide go in and out 1 trillion times. You would see the moon drift slowly back, the day grow slowly longer, the oxygen in the air increase fraction by fraction until you could breathe it. You would watch eventually the ice come, and you would watch it go. And in the ocean, you would watch something strange begin to happen. Cells forming associations, associations forming structures, structures developing specializations, specializations giving rise to bodies. You would watch the invention of the eye, the invention of the mouth, the invention of movement, not the chemical movement of bacterial flagella, but the muscular movement of an animal choosing its direction in the water. You would watch all of this happen over 1 billion years in absolute geological silence with nothing but the physics of the universe to explain it. It is the greatest story ever told, and it was told before there was anyone to tell it or to hear it or to understand it. We are the understanding. We are perhaps the universe's way of comprehending its own story. Rest now. The world is very old and it holds you. The oxygen in your lungs was made by life that has been breathing for 3 billion years. The carbon in your body has been living since before the continents existed in their current form. You are ancient. You are new. You are the inheritor of 1 billion years of work that was done without any thought of you. Sleep now in the long, patient silence of deep time. We return one final time to the shore. The light is changing. This shorter day moves faster than we are accustomed to, and the sun, already lower than it was an hour ago, is beginning to stain the sky with colors we might recognize, red and gold in the deep amber of coming darkness, because the physics of light scattering through the atmosphere does not change across 1 billion years, and a Proterozoic sunset looks, at least in this narrow register, like any other sunset. The same wavelengths, the same scattering, the same slow burning of the horizon. The tide is coming in. The moon is not yet visible in the darkening sky, but its pull is already moving the water, drawing it up the beach with its slightly stronger tidal force, covering the lower edges of the stromatolite beds with water that is warm. Warmer than the modern tropical sea at the same latitude in some estimates, and slightly different in chemistry, but still clear, still reflecting the sky, still moving in the rhythms of gravity that have not changed since the Earth first formed. The stromatolites are unaware of the sunset. They are unaware of everything in any sense we would recognize as awareness, but they are alive and they will continue. Tonight, as always, the photosynthesis will slow and then stop as the light fails, and the communities of cyanobacteria within the mats will shift into the chemical processes that sustain them through darkness. Metabolic pathways older than any other activity on the surface of this planet. The tidal water will rise and fall. The short night will pass and the sun will rise again sooner than we expect. No one is here to watch it. No one was ever here to watch it. And yet it happened. This shore existed, this sunset occurred, this tide came in and went out billions of times across hundreds of millions of years in absolute silence and absolute beauty, witnessed only by the mathematics of physics and the chemistry of life. There is something in that thought that the world was beautiful before there was any mind to call it beautiful, that I find worth sitting with, not because it diminishes beauty, but because it suggests that beauty, or something that would become beauty when finally perceived, was there all along, simply present. As the rocks are present, as the ancient light reaching us from a billion years away is present, encoded in the isotopic ratios of Proterozoic sediments, readable by instruments that are themselves the product of a billion years of evolutionary development that those same ancient rocks helped to make possible. We are the universe looking at itself. The cyanobacteria of the Proterozoic could not look. We can. And what we see when we look back far enough is not chaos, not randomness, but a deep continuity. A single planetary system changing always but never severing its connection to what it has been. The world you see when you open your eyes tomorrow morning, the sky, the trees, the wind, the particular quality of morning light through your window, is a world built on this foundation. Every atom of oxygen you breathe passed through something living before it reached you. The carbon in your body was once carbon dioxide drawn down by organisms not entirely unlike those ancient cyanobacteria on this Proterozoic shore. You are the long patience of the boring billion, finally arrived at consciousness, finally able to look back, finally able to imagine standing on a shore that no eye ever saw and find it beautiful. The world was old before we arrived. It will be old after we are gone. And in between we are here, the brief, improbable, extraordinary moment when the ancient shore finally became aware of itself, when the oxygen released by cyanobacteria a billion years ago found its way into a mind capable of wondering where it came from. That wonder is not a small thing. It is perhaps the most remarkable event in the history of this planet since life itself began. The universe folding back on its own past, recognizing itself across an incomprehensible distance of time. I wish you a calm and peaceful night. Let the deep time carry you into sleep. Let the ancient chemistry of the ocean's first oxygen drift through your dreams. Let the silence of a shore that no eye ever witnessed surround you like water, like the rising tide of the Proterozoic night. You are the heir to a billion years of patient work, and the world that made you was beautiful beyond all understanding, long before there was anyone to understand it. Rest now in the long patience of deep time. The shore is still there, beneath the sediment of a billion years, beneath the weight of all that followed. It remembers nothing, but it is there. And tonight, as you sleep, the oxygen it made is moving through you quietly as it has always done.